In this video, I will be presenting an experiment where we developed human reporter chondrocytes for SOX9 and PRG4. SOX9 is a critical transcription factor in chondrogenesis. PRG4 encodes the protein lubricin, which is an essential articular cartilage surface lubricant. The idea of this study is to produce a non-destructive and high throughput method of screening for compounds which may stimulate the promoters for both SOX9 and PRG4. Most chondrogenic analyses are destructive and limit temporal screening, so there's a need for a reporter cell like this to be developed. To accomplish this, we had a promoter-driven Gaussia luciferase plasmid that we put into HEK, human embryonic kidney cells, with lentiviral packaging and capsule plasmids. These lentiviral plasmids and our promoter gene formed a lentiviral vector which we used to infect human articular chondrocytes. These human articular chondrocytes became our reporter cells, and we had two cell lines, one for our SOX9 one for our SOX9 cells and one for our PRG4 cells. The idea is that in media, when the promoters are stimulated for SOX9 or PRG4, depending on which reporter cells we are using, it'll produce relatively more Gaussia luciferase protein. And using high throughput and temporal analysis, we can perform assays on this Gaussia luciferase protein, and we can get a luminescent response. To prove this model, we compared the SOX9 gene expression to Gaussia luciferase gene expression and PRG4 gene expression to Gaussia luciferase expression. We saw statistically significant correlation between these genes, and we showed a linear relationship. And looking at the luminescence, when stimulated by TGF-beta1, which is a known stimulator of both SOX9 and PRG4, we saw the increased luminescence values, showing that when chondrogenesis is stimulated, we're seeing an increase in our luminescent response. Histological analysis showed positive safranose staining, which indicates glycomine and glycans are still being produced in our reporter cells, showing we did not significantly alter chondrogenesis, helping support that idea. In this experiment, we also compared a vitamin and mineral optimized medias, both ATDC5 optimized media and a proposed theoretically optimized media, from the third paper referenced on this poster. We saw that the proposed theoretically optimized media did not increase either SOX9 or PRG4 promoter-driven luminescence. Temporally, we did see some increases in the ATDC5 optimized media for SOX9 and PRG4 gene expression. However, not all time periods showed statistical significance. This research enables us to monitor the expression of SOX9 and PRG4 in temporal 3D cultures without destroying the sample. And this will aid us in the development of new osteoarthritic therapeutics through temporal screening approaches. SOX9 and PRG4 expression in osteoarthritis were significantly altered in this study. However, the previously reported theoretically optimized media did not show an increase in promoter-driven luciferase expression for PRG4 or SOX9. Thanks for watching.